Assalamu alaikum students. Today I will tell you about how to make histological section of the male genital duct, that is the epididymis. Most of the students are afraid of making histology drawings, but after this tutorial you will see how it is easy to make a drawing of histological sections. Now, epididymis, it is a long, highly coiled duct surrounded by connective tissue that lies in the scrotum. You can see here. This is the epididymis. It is lying along the superior and posterior sides of each testes. It is about 6 to 7 meters long, and it begins in the head region of the epididymis and highly coiled in the body and then the tail, and at the tail end, in the tail region, it opens up into the ductus deferens. Now you can see here in the next picture. So this is the head, and this is the body, and this is the tail. You can see the efferent ductules from the ready testes are entering into the ductus epididymis. Okay. Now, if you have a cut section of the epididymis, now this is the cut section of epididymis, and you can see here this is the cut section of the epididymis, showing numerous profiles of the duct, which shows that the duct has a smooth luminal contour. You can see here these are the various profiles. Why? Because it is a single duct. Why there are various profiles? Because it is highly convoluted, highly coiled, and it is placed in a very narrow space. So when we cut it, we get multiple profiles, and you can see that the contour of the lumen is very much smooth. You can see in all these sections. Now, coming to the individual section of the duct of epididymis. Now, this is one section of the duct of epididymis, this is another, and this one is another. Now, you can see here that the duct of epididymis is lined by pseudostratified epithelium, and they bear long microvilli known as stereocilia on their free apical surface. The purpose of these stereocilia are to increase the surface area for absorption. Now, two types of cells are present in this epithelium, pseudostratified epithelium. Number one is the tall columnar principal cells, and number two are the basal cells. So tall columnar cells you can see here bearing the stereocilia, and basal cells lying in the basal parts. And both these cells are resting on the basement membrane. Now, in the lumen, you can see the stereocilia, long branching stereocilia, and the clumps of sperms. These are the clumps of sperms seen in the section of the lumen of the ductus epididymis. Now, regarding the functions of the epididymis, it is a temporary storage of spermatozoa. It serves as a conveying channels. It absorbs most of the water from the coming from the testicular secretion into the blood. It propels the sperms through the duct, and the cells also phagocytose some of the residual bodies and the degenerating spermatozoa, and also they secrete a factor known as decapacitation factors, which render the sperm infertile. Okay, now we will learn how to draw this histological section. This is the important part, and I think you will enjoy it. Now here, okay, first of all, when you draw a histological diagram, you have to draw it with very light hand initially. Then afterwards you can darken it so that at any moment you can rub and remake it again. So for drawing the section of this section of the epididymis, now in between the epididymis, duct of epididymis, lies the connective tissue, and outside the epididymis lies thin layer of smooth muscles which helps in propulsion of the, when they contract, helps in propulsion of the sperms. Okay, now first of all, try to make, okay, now I have drawn two sections, sections of the duct, cross sections of the duct. So this is the basement membrane. I have drawn it lightly, and then we will initially show the lumen, the smooth lumen. Okay, it will help us to draw the, you can see here I have drawn this, the lumen, the extent of the epithelium lightly here. So this is the lumen in between. Now we will draw the pseudostratified epithelium. Okay, so pseudostratified epithelium is drawn like that. So all the cells, they rest on the basement membrane, but not all the cells, they reach the apical surface. So this is how you define the pseudostratified epithelium. Okay. It's easy to draw at multiple sites and then join them, and drawing the cells at multiple sites, not from one side till end. So these are the basal cells, these are the columnar cells. You can see here I am drawing the columnar cells, basal cell, okay, the columnar cells, the basal cell, you can see here. Now you can fill in the various sites, so it's easy to draw. Draw with very light hand. Okay. So this is how you will draw the pseudostratified epithelium. Okay. columnar cells, the basal cells and the columnar cells. Sorry. Now we have finished making the pseudostratified epithelium, the cells of the pseudostratified epithelium. So you can see here like this. We will fill it now. Okay, so this is the basement membrane lying here and this is the lumen. Now we will fill it with the nuclei. So basal cells, so this is how we will fill the nuclei. So you can see here the stratification, pseudo stratification due to different levels of different positions of the nuclei of the epithelium. Here you can see the nuclei are lying at different levels, giving the false appearance of stratification. So this is pseudo-stratified epithelium, tall columnar pseudo-stratified epithelium with stereocilia. Now how will you draw the stereocilia in the lumen? With very light hand. So these stereocilia are long, so they combine with the neighboring cilia and giving the appearance of branching. So these are long microvilli. And they increase the surface area for absorption. So here you can see the stereocilia. Now we will draw the sperms inside the lumen. So how will you draw the sperms? We will just make heads first, head of the sperm containing the nucleus, many sperms. You can fill it with many sperms, many heads. And then the tails. So these are the clumps of sperms present in the lumen. So you can see here like that, the clumps of sperms present in the lumen. Now as I have told you before, the outer part of the this duct is covered by a thin layer of smooth muscles. So now we will make the smooth muscles outer to the. So for making smooth muscles, you only have to draw some lines. I will show you how to make these smooth muscles properly. Drawn some lines for the uh, smooth muscles, to form the smooth muscles. You know that smooth muscles are spindle shaped cells. Now we will draw their nuclei like that. Here you can see. Okay, now as you can see here, the smooth muscles, I have darkened them, and I have filled color in the middle part, while the ends are tapering, so you can easily make them like that. Okay, now this is the outer smooth muscle layer. This is the pseudostratified epithelium, stereocilia, and clumps of sperms in the lumen. Okay, now we will fill the cytoplasm in these pseudostratified cells. So coloring them a bit. Okay, so you can make it like that. So this is how you're coloring it. Okay, I will finish coloring it later. Now I'm drawing the fibrous connective tissue, which is lying outside the some blood vessels in the connective tissue. Fibrous connective tissue, irregular. So you can see here. So connective tissue fibers running irregularly in the blood vessels, and then we will draw the nuclei, endothelium in the blood vessels, venules, capillaries, arterioles, and the nuclei of fibroblast in the fibrous connective tissue. Okay, so this is one section. You can see here. Complete the coloring, so that the final picture you can see the final drawing, final diagram. So the basal parts. So this is how the so now you can see that this is the one section of the epididymis. Now you can label it, and this was the another. I haven't drawn it, but one section is enough if you have drawn it properly. Okay, thank you. Next time I will teach you how to draw the ductus epididymis and the testes, of course, and it will be coming very soon. Hope you enjoyed my tutorial, and please give comments and subscribe my channel if you like it.